The nerve impulse is discussed in the screencast, which may be found in Chapter 7 of your textbook. This screencast was designed to achieve the following objectives: describe a local potential that occurs in response to a stimulus, describe the production of a nerve impulse, describe how the plasma membrane is repolarized and its resting membrane potential restored after a nerve impulse is produced, describe saltatory conduction. In the previous screencast, we learned that at its resting membrane potential, the plasma membrane is polarized. There are more positively charged ions outside the cell compared to inside the cell, so there is an electrical gradient that will move positively charged particles into the plasma membrane. And if we measured the difference in charge across the plasma membrane with a voltmeter, it would register about -70 millivolts. The resting membrane potential, due to the unequal charges across the plasma membrane, contains potential energy like a charged battery, which is also polarized. In addition to the electrical gradient, we also have chemical gradients due to the higher concentration of potassium ions inside the cell relative to outside the cell, and the higher concentration of sodium ions outside the cell compared to inside the cell. Combining the electrical gradient and the chemical gradients, we could further describe the resting membrane potential resulting from an electrochemical gradient. First, I would like to outline the steps that are involved in the production of a nerve impulse or action potential by a neuron. Let us assume that this neuron, shown on the right side of your screen, has a plasma membrane that is at the resting membrane potential. A stimulus or change outside of the neuron will cause a depolarization in the plasma membrane. This depolarization is also called a local potential. This depolarization will spread from the dendrite and move toward the soma or cell body. You can sort of think of this spreading of depolarization like dominoes falling one beside another. You know how one domino hits the next, which hits the next, which hits the next, and it can go on for feet or yards or, I guess, potentially miles. That's basically what's happening as the depolarization spreads from the plasma membrane in the dendrites to the plasma membrane of the soma. As it spreads, it does lose intensity, but if it does reach the axon hillock, and it is of a sufficient magnitude, that is, if it exceeds what's called the threshold, it will cause a depolarization to spread from the axon hillock all the way down the axon to the ends of the axons called the axon terminals. And this spreading of depolarization from the axon hillock to the axon terminals, it's what's called a nerve impulse or action potential. Unlike the local potential, a nerve impulse or action potential is an all or nothing event. If it occurs at the axon hillock, it travels all the way down to the axon terminals and it does not degrade on its journey to the axon terminals. So that is a basic outline of how a neuron generates a nerve impulse. Now let's talk in detail about how this process occurs. Okay, so let's start at the dendrite. So we're looking at the plasma membrane of a dendrite at its resting membrane potential. So high sodium concentration outside, high potassium concentration inside, and there is a surplus of positively charged ions outside the cell compared to inside the cell at the dendrite. Recall that charged particles cannot easily cross the plasma membrane because the hydrophobic tails of the phospholipids don't allow charged particles to cross. The only way that charged particles cross is by some active process or if there are ion channels embedded in the plasma membrane. Now, there are sodium ion channels embedded in the plasma membrane, but at the resting membrane potential, they are closed for the most part. So sodium cannot cross. Now let's say along comes some type of stimulus. Now remember, a stimulus is nothing more than a change. It can be a change in light intensity, it can be a change in temperature, it can be a change in many variables. The stimulus that can be detected by a particular neuron really depends on the nature of that neuron. If the neuron is a photoreceptor in the eye, then it could detect a change in light intensity. If the neuron was an olfactory receptor in the roof of your nasal cavity, then it could detect a change in the concentration of molecules. Here, we're just talking about some generic stimulus. That stimulus, if detected by the neuron, is going to cause the sodium channels to open. Where the stimulus occurs, sodium channels open and sodium ions move down their concentration and electrical gradient into the cell. This depolarizes the membrane at the site of the stimulus. In other words, because positively charged sodium ions are moving into the cell, the difference in charge across the plasma membrane at that specific location is decreasing. This depolarization causes nearby voltage-sensitive sodium channels to open and allow sodium ions to move into the cell, which depolarizes the plasma membrane in that location, which depolarizes adjacent voltage-sensitive sodium channels, causing sodium to enter the cell, and so forth and so on. So we have this domino effect as sodium ion channels are opening farther and farther away from the original stimulus, and so we have this depolarization that is moving down the plasma membrane. And if we observed a voltmeter as that depolarization moved down the plasma membrane past the voltmeter, you'd see the voltmeter move towards zero or become less negative. So let's now summarize what has occurred up to this point. We have seen the generation of a local depolarization. On a dendrite, a stimulus has been detected causing a local depolarization to start to move toward the soma or cell body. We will represent the dendrite using this yellow bar here at its resting membrane potential. So here's the intracellular fluid, that's the cytosol, and the white space is the extracellular fluid. So a stimulus has occurred here. That stimulus caused sodium channels to open and for sodium to then move into the cell, causing a depolarization at the site of the stimulus, which we will represent using green. The depolarization occurring on the plasma membrane at the stimulus causes nearby voltage-sensitive sodium channels to open. As they open, sodium moves into the cell at that location, causing a depolarization there. That depolarization causes adjacent sodium channels farther away from the original stimulus that are also sensitive to voltage to open, allowing sodium to move into the cell. Thus, we have this domino effect as sodium channels open farther and farther away from the stimulus, and therefore we have this depolarization that is moving from the site of the stimulus down the dendrite toward the soma, and if strong enough, eventually to the axon hillock. I want you to note that a local depolarization does degrade as it moves farther and farther away from the stimulus. If we took a voltmeter and we measured the depolarization at the site of the stimulus, for example, it may register, and this is hypothetical, let's say it moved all the way from the resting membrane potential of -70 all the way up to, say, -15. If we moved farther away from the stimulus, however, we'd see that that depolarization was losing strength. Now it has moved to, say, -35, and then farther and even farther away from the original stimulus, it's almost back to the resting membrane potential. So the depolarization is losing strength as we move farther and farther away from the site of the original stimulus. So we have a local depolarization that was initiated by a stimulus on the dendrite. That depolarization is moving toward the soma, eventually to the soma, and if strong enough, it will reach the axon hillock. And as it moves away from the site of the original stimulus, it degrades or it loses strength, and I have illustrated that using the thinner and thinner arrows. However, if the depolarization, when it reaches the axon hillock, is of sufficient magnitude, that is, if it reaches the threshold, it will cause a depolarization to start in the axon hillock, and that depolarization will move all the way down the axon to the axon terminals without degradation, and that is called an action potential or nerve impulse. In other words, that local potential reaches the axon hillock, and if it is of a sufficient magnitude, if it's at or above threshold, sodium channels will open and the depolarization will move from the axon hillock all the way down the axon to the ends of the axons called the axon terminals. The same process by which the local depolarization spread from the dendrite to the cell body to the axon hillock is occurring throughout the axon. The main difference is that the nerve impulse or action potential does not degrade as it moves from the axon to the axon terminals. It is an all or nothing event. The analogy that your book uses is it's like flushing the toilet. If you push the handle far enough, the toilet flushes, and it flushes fully. If you recall, Schwann cells and oligodendrocytes form these coverings on the axons called myelin sheaths. These myelin sheaths do not allow sodium to enter the cell. They literally form a barrier where sodium ions are not available to pass through the sodium channels. However, if you remember, there are little interruptions in the myelin sheath called nodes of Revere. And here, sodium can pass through sodium channels into the cell. So as an action potential or nerve impulse moves down the axon, the nerve impulse appears to jump from one node to the next. And this is called saltatory conduction, and it does increase the rate at which an action potential moves down the axon. In the process of generating a local depolarization and then a nerve impulse, the plasma membrane of the neuron has been depolarized. That means it is no longer at the resting membrane potential and cannot respond to a further stimuli. In order to repolarize the plasma membrane, potassium channels open. Remember, potassium is higher inside the cell than outside the cell. So as potassium channels open, potassium ions are going to move from inside the cell to outside the cell, repolarizing the plasma membrane. And the potassium channels are going to open in the same sequence that the sodium channels open, from starting at the site of the original stimulus, basically following the same pathway of the depolarization. So the repolarization process is going to move from the dendrites to the cell body and eventually to the axon, repolarizing the plasma membrane. So after potassium channels open and repolarize the plasma membrane, we may have restored the electrical gradient across the plasma membrane, but notice that the sodium and potassium concentrations have switched sides. We now have a higher sodium concentration outside the cell and a higher potassium concentration inside the cell. That's where the sodium-potassium pump comes in, moving 2 potassium ions into the cell. For every 3 sodium ions, it moves outside the cell, restoring the resting membrane potential, where the concentration of potassium ions is higher inside the cell and the concentration of sodium ions is higher outside of the cell. Let's review the objectives of this screencast. Describe a local potential that occurs in response to a stimulus. Describe the production of a nerve impulse. Describe how the plasma membrane is repolarized and its resting membrane potential restored after a nerve impulse is produced. And lastly, describe saltatory conduction. The next screencast discusses a chemical synapse.